That's right. This has the potential to end blood shortages if all goes as researchers hope. There are 4 blood types, and I'll walk you through exactly how the researchers say this could work.
Blood type O was supposed to be the boring one, the blank, the default. So when researchers fed millions of genomes and millions of medical records into machines built to find patterns no human could see, nobody expected the letter O to say anything at all. They were checking a box, ruling it out. Instead, the models kept flagging it, again and again and again, until the signal was too loud to explain away. What they were looking at wasn't a medical detail. It was a message, and it had been sitting inside almost half the human race the entire time. One letter.
O negative is especially needed in trauma situations where emergency room doctors immediately jump into action.
It's one letter, just one. It sits in a small box on a donor card, or buried somewhere in a medical file most people never open twice. For a huge share of the human population, that letter is O. Most people last thought about it in a clinic chair while a nurse pressed a cotton ball to their arm and told them to keep pressure on it. It's the kind of thing that comes up at a dinner party when the conversation stalls. It feels about as meaningful as a shoe size. But that single letter is older than farming. It's older than written language. It's older than cities, older than agriculture, older than every myth we've ever told about where we came from.
Maybe you've heard of the blood type diet, maybe you haven't. But in this video, all we're gonna talk about is the O blood type.
In a very real sense, it's older than our species. The variation in human blood that earns a person that O has been carried, copied, and quietly preserved across an almost unimaginable stretch of time, long before anyone thought to draw a vial and give it a name. And when researchers finally did what modern science does best, when they stopped asking what blood type means and started asking what it correlates with, at a scale no previous generation could have processed, the plain default, the nothing special option, refused to behave like a label. To understand what the models found, you first have to understand what they were looking at, because most people carry the letter without ever knowing what it stands for. What type O actually is.
There's 47 different blood group systems, and within those 47 blood group systems, there are over 300 different antigens that make each of us unique.
Blood is made of a few basic ingredients: red cells, white cells, plasma, platelets. It's the red cells that give us the system, the classic 4-way split into A, B, AB, and O. And the difference between them isn't the color of the blood, or the thickness of it, or anything visible to the eye. Put 4 vials on a table from 4 different donors, and no one could tell them apart. The difference lives on the surface of the red blood cells, in the form of tiny molecular markers called antigens. Think of them as little flags planted on the outside of each cell. Flags the immune system uses to tell friend from foe. Cells flying the A flag make a person type A. Cells flying the B flag make them type B. Cells flying both make them the rarer type AB, and if they fly neither, no A flag, no B flag, a smooth surface with the marker simply absent, that person is type O. That is the whole secret of O. It is defined not by what it has, but by what it's missing. In older medical language, O sometimes gets called the null type, the blank, the empty box, a category built out of nothing, which sounds like a footnote. It is not a footnote. That blankness is the reason O behaves the way it does in an emergency room. And it is the reason, as the models would eventually suggest, that O behaves the way it does across 10,000 years of human history. The universal donor. Anyone who has donated blood can already feel where this goes. Those flags, the A and B antigens, are exactly what the immune system attacks when it meets blood it doesn't recognize. Give type A blood to someone who is type B, and their immune system sees unfamiliar A flags, sounds the alarm, and launches a reaction that can be dangerous, even deadly. That's why blood has to be matched so carefully before a transfusion. It isn't bureaucracy, it's biology with a very short temper. But O blood, the blank type, the one with no A and no B flags to raise the alarm, slips through. There is nothing on its surface for a stranger's immune system to reject. Which is why, in one particular form, O blood earned a title that sounds almost heroic: the universal donor. Picture the scene it was built for: an emergency room in the middle of the night. Someone comes through the doors after a crash, losing blood fast, unconscious, unable to say a word about who they are, let alone what type runs in their veins. There's no time to draw a sample, send it to a lab, and wait for an answer. Every second is measurable in tissue running out of oxygen, so the staff reach for the one thing they know is safe to give to almost anyone. Bags of O-negative blood, kept ready, kept cold, kept stocked precisely for the moments when there's no time to ask questions. That blank, flagless blood becomes the thing that keeps a stranger alive long enough for the rest of medicine to catch up. It has done this quietly, in hospitals all over the world, for decades. Most of the people it saved never learned the letter that saved them. So already O is doing more than sitting in a box on a card. But the emergency room is the part everyone knows. What almost nobody knows is where that blankness came from. And the answer runs directly against the assumption nearly everyone makes. The mutation that won.
Back at home this morning, a team at the University of British Columbia says it has found a way to transform blood and convert all types into the universal type.
The instinct is to assume O came first, that it's the simple one, the blank one, the starting point, and that A and B are the elaborate add-ons that showed up later. The original model, with everything else an upgrade, it's the opposite. The best current understanding is that O is the newcomer. Deep in the human genome sits the gene that builds those antigen flags. And at some point in the distant past, in some ancestor whose face will never be recovered, a piece of that gene's instructions broke. A small stretch of code was lost. With the instructions damaged, the cell could no longer finish building the flag at all. The result was a red cell with a blank surface. The very first type, OO, isn't the original. O is the mutation, the variant that arose when the machinery for making antigens quietly failed. And then that failure spread. It didn't die out. It didn't stay rare. It didn't linger as a curiosity in one isolated valley. It multiplied. It crossed continents. It rode along with every migration humans ever made until it became the single most common blood type on the entire planet. A broken gene became a winner, which raises a question that sits underneath everything that follows. Why would a variation that began as a loss, as damaged instructions, as an absence, go on to outcompete the very thing it was missing? Nature does not usually reward broken machinery at that scale, for that long, unless the breaking buys something in return. A quick word. This channel exists for exactly these questions. The hidden histories written into the human body, the things we carry without ever being told what they are. If that's the kind of story you want more of, subscribe and stay with us. There's a great deal still buried in here. Now, what did the breaking buy? To answer that, you have to look at where in the world type O shows up, and where it very nearly takes over completely. The map. Look at a map of human blood types, and something jumps out immediately. Type O is not spread evenly across the world. It clusters. It surges in some places and thins out in others. And those patterns are not random noise. They are the fingerprints of history, of migration, of isolation, and, as the analysis would eventually suggest, of disease pressing down on our ancestors for thousands of years. Start in the United States, where roughly 45% of people carry type O in one form or another, already close to half the population, which tells you how successful this so-called broken gene has been. But 45% is a moderate number on the global scale. Travel south, deep into parts of Central and South America, and the figure doesn't just rise, it climbs toward the ceiling. In certain indigenous populations, particularly in regions that stayed isolated for long stretches before the arrival of outsiders, the frequency of type O approaches something that sounds almost impossible, near totality. Whole communities where practically everyone shares the same flagless blood, and where the A and B antigens, so common elsewhere, had all but disappeared from the mix. When a single genetic variant reaches that kind of dominance in an isolated population, geneticists take notice, because it usually points to one of two explanations, and telling them apart is genuinely hard. The first is a bottleneck, a founder effect. Imagine a small group, a few dozen people, perhaps fewer, breaking away from a much larger population and crossing an enormous distance. A land bridge, a coastline, a route no one had walked before. Whatever mix of blood types existed in the population they left behind does not travel with them intact. Only the types those particular travelers happen to carry make the journey. If, by pure accident, most of that founding group carried O, then every descendant population born from them starts out overwhelmingly O. Not because O was better, not because anything selected it, because of who happened to be standing in that group on that day. Then compound that over generations. Add hardship. Add further splits, further small groups peeling off into further valleys. Each split narrows the deck again. Chance, amplified by isolation, gets frozen into an entire people. That is the first explanation, and it involves no advantage at all. It is history, not biology. The second explanation is selection, that something in the environment was actively favoring O and pushing the alternatives out. That the people carrying A and B were, for some reason, marginally less likely to survive long enough to have children in that particular place. The two explanations look almost identical on a map. Both produce high O frequency. Both produce clustering. And for most of the 20th century, arguing between them was largely a matter of taste, because there was no way to test one against the other at scale. In truth, both forces almost certainly played a part, but which one, and how much, and where, that question sat unresolved for decades. Now, swing the map the other way, across the oceans to Asia. Here the picture shifts again. Type B, comparatively uncommon in much of Europe and the Americas, becomes far more frequent across broad stretches of Central and East Asia. There are regions where B is woven through the population in a way that would look almost exotic to someone from Peru or Ireland. And in parts of the Indian subcontinent, type A rises to prominence in certain communities, holding its ground against the others. No single type wins everywhere. Instead, there is a shifting mosaic. O dominant here, B climbing there, A leading in a third place, as though the species had been dealt slightly different hands depending on where its ancestors happened to walk. That mosaic is the thing to sit with. If blood type were a meaningless label, trivia with no consequences, it would be scattered evenly, spread thin and random across every group, the way a trait with no stakes tends to be. It isn't scattered evenly, it's structured. It follows the contours of continents and climates and something else, something invisible, the ancient geography of disease, which sharpens the question into something specific enough to actually test. What was it about certain environments that, quietly, generation after generation, tilted the odds toward the blank-flagged blood? Answering that required a tool nobody had until recently. Not a name tag. First, a correction to the way most people picture an antigen. It is not a name tag. Those flags are real physical structures, sugars built onto the surface of red cells. They don't merely identify the cell, they interact with the world. Bacteria can grab onto them, viruses can use them as handholds, parasites can recognize them, exploit them, or be blocked by them entirely. The surface of a red cell is not a blank billboard with a letter printed on it. It's terrain, a landscape that other living things are constantly trying to read, climb, and use. And here the flagless nature of type O stops being a curiosity. If a microbe evolved to latch onto the A antigen, a person whose cells carry no A antigen offers that microbe nothing to grip. The very absence that defines type O can turn a red cell into a smooth, featureless, unwelcoming surface for a specific invader. What looks like a defect becomes, under the right pressure, something closer to armor, not against everything, never against everything, but against particular threats in particular places. The blank surface could mean the difference between an illness someone recovers from and one they don't. Follow that logic forward. If a deadly disease was common in a region, and that disease worked better against people carrying A or B, then over enough generations the survivors would skew toward those whose blood gave the disease nothing to hold. Their O would pass down. Their neighbors A and B would thin. Across centuries, the map would slowly redraw itself into exactly the pattern we see, elegant, testable in principle, and, for a very long time, impossible to prove. Because you cannot run an experiment on the deep past. You cannot expose two matched populations to a plague and see which one walks away. History does not sit still for a control group. What you can do instead is read the survivors, all of them at once. What the machines did. This is the part the title is pointing at, and it deserves plain language rather than mystique. Over the past 2 decades, research institutions assembled something that had never existed before: biobanks. Enormous repositories linking anonymized genetic data to electronic health records. What conditions a person developed, what medications they took, what happened to them over decades. across hundreds of thousands or millions of participants. On its own, that data is unreadable. No human being can hold 1 million genomes in their head, let alone cross-reference them against 1 million medical histories. So, researchers turned to machine learning models and genome-wide association analysis, methods designed to sweep across the entire genome, one variant at a time, hunting for statistical associations between a genetic difference and a health outcome. The models don't know what a blood type is. They don't know what malaria is. They have no theory to defend and no hypothesis to protect. They look for signal in a haystack of noise, and they flag whatever survives statistical correction. That indifference is the point. A human researcher looks where they expect to find something. A model looks everywhere. And when that machinery was pointed at the ABO region of the genome, it did not return a shrug. The first thing it returned pointed toward one of the oldest and deadliest enemies our species has ever faced. A disease carried on the wings of a mosquito, one that has killed more human beings across history than almost anything else. Malaria, the oldest enemy. That disease is malaria. To understand why it matters here, sit for a moment with how relentless it has been. For most of human history, in the warm and wet parts of the world, malaria was not a rare tragedy. It was a constant, a tax, a season. It thinned villages. It killed children before they could walk. It returned year after year, generation after generation, pressing down on entire populations with a weight measured not in decades, but in millennia. When a threat is that heavy and that patient, it does something to a species. It leaves fingerprints in the genome. Anything offering even a slight edge, even a 2 or 3% better chance of surviving a severe bout, would slowly, quietly, over countless lifetimes, become more common. Not dramatic, not fast, just arithmetic running for 10,000 years. So, the analysis went looking. Health records and blood type data from regions where malaria had long been part of daily life, compared against places where it never took hold. Not dozens of people, not hundreds. Datasets cross-referenced at a scale that would have been impossible a generation ago, and a soft but stubborn signal appeared. It was not that people with type O never caught malaria. They did constantly, everywhere it existed. It was that among those who fell ill, the ones carrying type O appeared, on average, somewhat less likely to develop the most dangerous form of the disease, the severe kind. The kind that fills the smallest blood vessels, shuts down organs, and turns an illness into a funeral. Type O seemed modestly to soften that worst-case slide. And this is exactly the kind of finding that gets twisted into something it isn't. It does not mean a person with type O is safe from malaria. It does not mean anyone should feel protected, skip a mosquito net, or take a single risk they wouldn't otherwise take. The effect is small. It's a nudge, not a shield. It appears only when you zoom out across hundreds of thousands of people, the way a faint pattern in sand only resolves from a great height. At ground level, looking at one person, it is invisible. But zoom out far enough, across whole populations and whole centuries, and a nudge that small can still bend the shape of a species, which left the obvious follow-up question hanging. A correlation is not a reason. Was there an actual mechanism, or was this just a coincidence buried in a very large spreadsheet? The mechanism. There appears to be a mechanism, and it lives in the physical structure of the cell. In severe malaria, infected red blood cells can clump together with healthy ones, forming sticky clusters, rosettes, that jam the tiniest blood vessels and starve tissue of oxygen. That clumping is a large part of what makes severe malaria lethal. It isn't only an infection, it's a traffic disaster inside the circulatory system. And the work suggests those rosettes tend to form more readily and hold together more tightly when the red cells carry A or B antigens. On the smooth, blank surface of a type O cell, they appear to form less easily and fall apart faster. So the very absence that defines O may have made the difference between blood that kept flowing and blood that jammed itself into a crisis. Now, put that beside the map. Type O climbing toward near totality across parts of South America. Type O running high through swaths of Africa and other tropical regions. Overlay that against where malaria historically raged, and the two pictures rhyme. Not perfectly. Nothing in biology is perfect, and other forces shaped these populations. Migration, isolation, founder effects, sheer chance. But the echo is there, quiet and persistent. Exactly the echo you'd expect if an ancient disease had spent thousands of years gently thinning one blood type and sparing another. That was the first crack in the idea that Blood type is nothing more than a label on a donor card. And once that crack existed, there was no reason to stop at one disease. The clotting thread. If the absence of A and B mattered for a parasite, the same logic ought to apply anywhere those antigens touch the machinery of the blood itself. So, the lens widened, the same large-scale comparison, this time run across clotting. The conditions where blood thickens and jams when it shouldn't, where a clot forms somewhere it has no business forming, or breaks loose and travels somewhere it can do terrible harm. Another modest signal surfaced. People with type O tended on average to carry slightly lower levels of certain proteins involved in the clotting cascade, and lower levels of those factors correlate gently with somewhat reduced risk of some kinds of clots. The caution here has to be stated differently because the temptation is different. Nobody should read this as a verdict on their own body. Clotting risk is built from a tangle of factors vastly larger than blood type: age, mobility, surgery, injury, medication, pregnancy, inherited clotting disorders of entirely separate origin, and plain luck. Against all of that, type O is one faint thread in an enormous tapestry, and pulling on it alone tells you nothing useful about any individual. But it is a real thread. Read enough people, and it shows up. And it obeys the same strange logic as the malaria finding. The notion that this null, this so-called defect, comes bundled with quiet consequences rippling out into the body in ways nobody chose and nobody designed, which is where a comfortable story would end. Broken gene turns out to be secretly useful. Everyone goes home satisfied, except the data refuses to end there, and the reason it refuses is written in the survival of every other blood type on Earth. The cost. If type O were purely an advantage, if the absence only ever helped, evolution would have pushed nearly everyone toward it long ago. A and B would have thinned out and vanished, the way losing variants generally do. They didn't. A and B are still very much with us, still common, still passed down, generation after generation, on every continent. That fact alone is an argument. It means type O must carry costs, and the reason it carries costs comes back to what an antigen actually is. A pathogen doesn't attack blood in general. It attacks a specific molecular shape. Evolution optimizes it toward whatever surface it encounters most often. Some microbes evolve to grip the A structure, some exploit the B structure, and some, over long enough exposure, become better adapted to the surface where those structures are absent entirely. There is no such thing as a cell surface that is invisible to everything. Removing a handhold removes it only for the organisms that were using it. For the rest, the blank surface isn't camouflage. It's simply a different kind of ground, and something out there is adapted to that ground too. That prediction turns out to be visible in the record. The analysis hinted that people with type O may be somewhat more susceptible to certain stomach ulcers, the kind driven by a particular bacterium that appears to find the O surface a little more hospitable for colonization than the alternatives. Where the antigens are absent, this organism seems to establish itself slightly more comfortably against the stomach lining, and the inflammation that follows is what eventually becomes an ulcer. And in the case of cholera, the violent, dehydrating illness that has swept through human populations again and again, and killed with terrifying speed, some large-scale analyses suggest type O may be linked to more severe outcomes rather than less. Not more infections necessarily, but worse ones. Faster fluid loss, harder crashes. So, the same blood that may soften the worst of malaria might sharpen the worst of cholera. The armor that turns one enemy away holds the door open for another, which is why this letter refuses to sit still as a simple label. It isn't good or bad. It isn't better or worse. It's a set of quiet bargains struck over unimaginable stretches of time, in which every gain arrives tethered to some loss. And that balance, that stubborn refusal of any one type to win outright, is the thread that, when the analysis pulled on it, led somewhere nobody had scheduled. Because the next question was not about disease at all. It was about how long this argument has been running. Older than US. The obvious way to date a genetic variant is to compare it across species. If a variation exists only in humans, it arose after our lineage split from its nearest relatives. If it exists in relatives too, it's older than the split. So, the comparison was run, and the result is the moment the story stops being about medicine. The same basic variation, the presence or absence of these antigens, shows up not only in humans, but in chimpanzees, in gorillas, in other primates on branches of the family tree that separated from ours millions upon millions of years ago. This is not a trait our species invented. It's an inheritance we walked in carrying. handed down through an almost incomprehensible chain of ancestors, most of whom would look nothing like a modern human. Ancestors who never built a fire, never spoke a word, never buried their dead, never once looked at their own blood and wondered what was in it. And the persistence is the real puzzle, a much stranger puzzle than malaria ever was. In evolution, most variations do not last that long. They rise, they fade, they get swept away as one version proves useful enough to crowd the others out. That is the ordinary course of things. Variation is temporary. Winners emerge. Populations settle. This variation has refused to settle across species, across ice ages, across the rise and collapse of countless populations, across the total redrawing of the planet's climate more than once, across every migration our lineage ever attempted. All these types kept surviving side by side for a span of time that dwarfs the entire existence of anatomically modern humans. Biologists have a name for that kind of stubborn coexistence: balancing selection. It's what happens when no single option is permitted to win, because each one is good at something and bad at something else, and the environment keeps changing its mind about which trait it rewards. Follow the implication. If type O helps against one disease and hurts against another, and type A does roughly the reverse, and the diseases themselves ebb and flow across regions and centuries, arriving, raging, retreating, returning, then no type ever gets to dominate for long. A cholera outbreak might quietly disadvantage the O carriers for a generation. A wave of malaria might quietly favor them for 10. A migration reshuffles the deck entirely. A change in climate moves the mosquitoes north, and the whole calculation inverts. And over deep time, the population never resolves. Not because the pressure is weak, but because the ground it stands on never stops shifting. The blood in your veins is a snapshot of a negotiation that has never concluded, and probably never will. This is the part the models lit up that no folklore ever could, and the part researchers had the least framework ready for. Balancing selection cannot be seen in a single patient. It produces no symptom, no sensation, no measurement any physician could take. It leaves no mark on a chart. It exists only as a shape in aggregate, visible when you read across enormous numbers, across whole populations, across species, across geological time. A finding of that kind cannot be discovered at a bedside. It can only be computed, and when the scale finally became vast enough for those slow, patient trade-offs to rise out of the noise, the letter on the donor card, the most forgettable fact in a person's medical file, turned out to be a fingerprint of one of the oldest ongoing experiments in biology, still running, still unresolved, printed into the body of very nearly every living human being, which makes the second half of what the analysis produced almost harder to accept. Because the same machinery that handed us something, this old also took something away. What the data took apart. For every genuine finding buried in that data, there were popular beliefs about blood type that the same analysis quietly dismantled. Start with the diet. Somewhere along the way, an appealing story took hold, that each blood type traces back to a certain kind of ancestor, and that a person would thrive by eating the way that ancestor supposedly ate. Type O, in this telling, belonged to the hunters. Meat, protein, the food of a lean and ancient forager. It's a seductive idea because it borrows the emotional weight of something true, the genuine antiquity of these variants, and attaches it to something invented. That's exactly why it spread so far. But the idea makes a testable prediction, and the prediction is specific. Matching food to type should produce a benefit that mismatching does not. When that was tested, the prediction failed in a very particular way. People following those diets did often improve. Their markers moved. They felt better. That part was real, and it's why the belief survives. The problem is what the improvement tracked. When participants were sorted by their actual blood type, the benefit didn't follow the match. People eating the so-called type O diet improved whether or not they were type O. The gains tracked the diet itself. More vegetables, less processed food, more attention paid to eating at all. And they appeared just as readily in the people who were, by the theory's own logic, eating the wrong food for their blood. Swap the types around and the results hold, which means the letter was doing no work whatsoever. It was a label attached to a change that would have worked regardless. The personality claim collapsed in a different way, and the way matters. In some parts of the world, the belief that blood type shapes character runs genuinely deep, woven into casual conversation, into matchmaking, sometimes into how people size each other up before exchanging 3 sentences. Type O as the natural leader, the fighter, the one built for the front of the pack. As culture, it's fascinating. It's an entire shared vocabulary for talking about who people are. As science, it doesn't survive contact with scale. Small samples produce apparent correlations constantly. That is simply what small samples do. Measure enough personality traits across a few hundred people, and something will look significant by chance alone. And when a culture already believes the association, two more effects pile on. People report themselves in ways that fit the label they have carried since childhood. And researchers who expect a pattern find one. Increase the sample size and those effects wash out. The correlations shrink towards zero rather than sharpening. Nothing replicates. Across large groups, no trait, no temperament, no measurable disposition attaches to the antigens. The stories remain meaningful to the people who tell them. The blood itself has no opinion about who anyone is. And that split, durable findings on one side, dissolving myths on the other, is the whole lesson compressed. The analysis was generous with truth and merciless with fiction. It handed over malaria and clotting and cholera and a piece of evolutionary time older than our species. And in the same motion, it retired the diet charts and the personality readings, not out of cynicism, but because they simply were not there when anyone looked closely enough. Which leaves one question. What is a person actually holding, standing here with this letter? What you walk away with. Not a diagnosis, not a prophecy, not a meal plan or a personality profile. What you hold is a single faint thread in an enormous weave, one small factor among thousands that shape a human life, and nearly all of them louder than this one. Your blood type nudges a few probabilities across, a whole population. It does not decide your health, and it certainly does not decide your fate. And that distinction matters practically. Anyone with a real question about their own body, their own risks, their own choices, belongs in a conversation with a doctor who knows them and knows their history, not staring at a letter, trying to read a future in it. This is the wonder of the wide view. It is not advice for the single case, but the wonder is real, and it's worth sitting with. Because that forgettable mark, the thing you barely think about after leaving the clinic, the thing filed somewhere between shoe size and postcode, turns out to be one of the oldest sentences still being written in the human body. It reaches back past history, past agriculture, past our own species, into a negotiation with disease that began before we had faces anyone would recognize, and hasn't finished yet. It doesn't tell you who you are. It tells you where you came from, in a language older than any story we invented to explain it. They fed the machines everything, expecting a shrug. They got a message from a few million years ago instead. So, now I want to hear from you. Do you know your blood type? And be honest. Did you ever believe one of the myths? Did you try eating for your type? Did you grow up somewhere that read personality into it? Or did you just assume O meant nothing more than a line on a donor card? Tell me your type, and tell me the belief you let go of, down in the comments. I genuinely want to see how many of us were carrying the same stories, and if this changed the way you look at that one small letter. Subscribe and stay with us. There's a whole hidden history written into the human body, and we are only beginning to learn how to read it.